Good morning, everyone. Hello, good morning. How are you all doing? Good morning, Anup. Good morning, Sajida. Good morning, Pooja. Good morning, Kirtana. Good morning, everyone. So, how are you all doing? I hope everyone is doing fine, right? Yes, good morning. So, yesterday you dealt with Unit 7. So, today I have come up with Unit 8, that is genetics, in which you'll be studying about Mendelian genetics. So lot many questions comes from Mendelian genetics, numerical based questions related to probability and all that stuffs, which students usually find very much difficult. So we will be clearing all our concepts here related to Mendelian genetics alone. So there is extension of Mendelian genetics also there, but we'll be not covering that part. Today we'll be covering with Mendelian genetics only, right? So the discussion for the topic for the discussion today would be regarding Mendelian genetics, where we'll be studying about the comparison between genotype and phenotype. Means people know like what is genotype and phenotype, yet knowing the comparison between both of these terms is very important, because it is important to understand the concept in which the questions are being framed, and if we are clear with these concepts, we can easily solve those particular questions related to probability and what the question is really asking, right? So Mendel's experimental design will be studying, just an overview will be going through. Then the comparison between the homozygous and heterozygous genotype. Then we'll be studying about the monohybrid crosses and its conclusion that was being made by Mendel, and the comparison between the test cross, back cross, and reciprocal cross. What are these crosses? What are their significances? And how do you recognize in a question when a test cross is being given to you or when a reciprocal cross is being given to you, right? And on that basis, how can you solve a particular question? All that thing we'll be discussing today. Then comes the dihybrid cross and its conclusion, and the Mendelian genetics formulae that is basically being used here, right? So here was the question of earlier session of Unit 6, yes, photosynthesis, where I had halted upon these two particular questions. I hope everyone would have solved these questions, or else we'll be solving here, right? So the first question is regarding the PS1 and PS2, the photosystem 1 and photosystem 2. And we compared about both of these photosystems in our earlier sessions, right? So I hope everybody would have gone through the standard books and tried to solve these questions. So first question says that the light reactions of the photosynthesis are carried out by four major protein complexes: photosystem 1, photosystem 2, the cytochrome B6 complex, and the ATP synthase. The following are the certain statements on PS1. So there are the statements given on PS1 that you have to recognize which statement is correct, right? So PS1 reaction center and the PS2 reaction center are uniformly distributed in the granule lamellae and the stromal lamellae. Second says the electron donor for the P700 of PS1 is plastocyanin, and electron acceptor of P700 excited form is a chlorophyll known as A0. Third, it says that the core antenna and the P700 are bound to two key proteins, PSA-A and PSA-B. That is also something we have studied and came across into that revision table that we have compared between PS1 and PS2, right? Then it says that the cyclic electron flow occurs from the reducing side of the PS1 via the plastohydroquinone and B6F complex. This supports the ATP synthesis but does not reduce the NADP+. So which of the following statement is correct according to you? Yes, I'll be inviting the answers. Yes, Ankita, video has started. So we are discussing the earlier questions that we discussed in our earlier session of Unit 6. So these are the two questions that you need to answer. First question is majorly related to PS1. Anybody, if you would have solved these questions? So Babita, for free sample papers, what you can do is there would be like a link that would be given at the end of the session. That link would be regarding the Telegram group in which these sample papers, free sample papers would be posted. So from there, you can download the free sample papers, right? So at the end of the session, you can download it. So Vishal says it's 4. Okay. Sudhakar 3. Keethna 3. Okay. So majority are saying 3, right? So why do you think option D is correct and option C is not correct? Because we have studied when we compared regarding PS1 and PS2 that the core antenna and the P700 are bound to 2 key proteins, PSA-A and PSA-B. That we have studied, and C is the correct option here. And in 3 option, there is no C statement which is coming. So 3 option is not correct. So here the option 1 is correct. Now let me tell you how, right? Option 1, how it is correct. The first statement, it says that the PS1 reaction center and PS2 reaction centers are uniformly distributed in the granule lamellae and the stromal lamellae, right? So both in the unstacked and the stacked region of the thylakoid membrane, both of these PS1 and PS2 are uniformly distributed. First statement is correct. Second, it says the electron donor for the P700, that is not in the excited state of PS1, is plastocyanin, right? So from plastocyanin, the electron is being donated to P700, and the electron acceptor of P700, when the P700 gets an electron, into the excited state, then it transfers its electron to the chlorophyll known as A0. This is also the correct statement. The order of the electron transport, which was taking place in the Z scheme, right? C is the core antenna and the P700 are bound to the 2 key proteins. Yeah, this one too we studied in the comparison table itself, the Conceptica table that I shared with you, people, the PS1 and PS2 comparison table. In that, this statement was written. If you can go through the earlier session, you can find this, right? Then D statement says that the cyclic electron flow occurs from the reducing side of the PS1 via the plastohydroquinone and B6 F complex. This supports the ATP synthesis. So last statement is correct that it supports ATP synthesis but does not reduce the NADP plus. But the earlier statement says that the cyclic electron flow occurs from the reducing side of PS1. No, like electron flow basically occurs from the oxidizing side, because PS1, when it transfers, it gets oxidized, right? It doesn't get reduced. So this particular statement, statement D, is incorrect. So all these 3 earlier statement A, B, and C are correct. So regarding that, we can see that option 1 is the correct option here, right? Now coming to the second question, which says that which one of the following is the correct order of electron transport during light reaction in the thylakoid membrane of chloroplast. Now this is related to Z scheme, right? This is the electron transport order that you have to, you need to be thorough with. Now, which one of the following options would be correct? Yes, I'll be inviting the answers. Okay, video zone, he says 4. Anup is 4, yes. Correct option is 4. Okay. So majority of the people, you are saying that correct option is 4, and you are absolutely correct. So option 4 is the correct one. The electron is being transferred from P680, that is the PS2, right? And it is being transferred to the plastoquinone, then it is being transferred to cytochrome B6F, and at last it is being transferred to plastocyanin, right? So this option is the correct one. So good. Now moving on to our unit 8. So here, with the help of the comparison table, we are comparing between the allele and trait. What is the difference between allele and trait? So here it says that the allele, an allele refers to the one of the 2 or more alternative forms of genes. So this is something basic which everybody knows, that alleles are basically the alternative forms of genes, right? Yeah. So as we have studied in case of Mendelian genetics, there will be tall, this is the homozygous dominant one, this is the homozygous recessive one. So both of these are basically the two forms, right? Both of these are basically the two forms of one gene itself, right? One gene which is responsible for the height. So for height, there are two different alleles. It could be capital T or it could be small t. So these are known as the alleles. Now, trait is basically refers to the genetically determined characteristic that belongs to a particular individual, right? So these alleles, they will basically decide the trait of a particular individual. So if we talk about this tall character, so this is the heterozygous form, right? So for this, the offspring which would be produced, it will have the trait as tall, right? Because this is the dominant one, this is the recessive one, and as we all know from the law of dominance, the recessive trait, it is being masked by the dominant trait, right? So this is basically, the tall is basically the trait of the individual. So these alleles basically define the trait of an individual, right? So allele, as we know, it is an alternative form of a gene, right? And the character, that is the trait, it is being defined or determined by these alleles. Then it says that also called the genotype of an individual. So allele is basically determines the genotype of an individual. So capital T, small t, it would be heterozygous, right? Heterozygous dominant, and this would be the genotype of an individual, right? Then it says that also called the phenotype, for the trait it says that also called the phenotype of an individual. So trait, it basically represents the phenotype of an individual, like what an individual really looks like from outside, right? It is a physical character actually. Then allele, it is located on the same loci on the homologous chromosomes, right? Two homologous chromosomes from both the parents. This is one chromosome, this is the second chromosome. So here would be capital T in male parent, in female parent, and here would be the small t in case of the other parent, right? So these are the two homologous chromosomes in which in the same loci, these two different alleles are present for the same character. The character remains the same right here. Then it says that can be visualized through the DNA testing. This alleles can be visualized through the DNA testing, and in case of phenotype, most are visible through our naked eye. So for visualizing the alleles, the genotype, we have to do the DNA testing. It is not visible through our naked eye. While the traits or the physical characters of an individual, they're visible for through the naked eye. And then it says that always occurs in pairs. So alleles always occurs in pairs. It is never occurring in a singular form, right? And traits, they occur individually. Individual to individual, the traits differ. It is not being influenced. So allele is not being influenced by the environmental factor, right? Only the traits, that is the physical character of an individual, it is being influenced by the environmental factors. So then we'll be understanding what is genotype and phenotype, right? This is regarding same only. So genotype is related to allele only. In different form we are studying here, and phenotype is related to the trait, right, or the character. So here it says that genotype is the genetic constitution of an organism, right? So genotype, it is a genetic constitution of an organism. Phenotype, it is observable trait. So this trait is basically being observed through the naked eye, and it differs from individual to individual, or a set of traits, whether it is structural form or the functional form of an individual, produced by interaction between its genotype and the environment. So both genotype and environment plays a major factor here for deciding the phenotype of an individual, right? Then here it comes the relationship between the genotype and phenotype, how genotype affect phenotype, right? So as we have studied in Mendelian monohybrid cross, what do we do? Like in case of monohybrid cross, the genotypic ratio that we study is basically, genotypic ratio that we study is 1:2:1. That is what we do, right? So 1 is basically the homozygous dominant one, This one is basically the this heterozygous dominant, and this is basically the homozygous recessive. So this is the genotypic ratio which comes. So this genotypic ratio basically defines the phenotypic ratio. So phenotypic ratio is dependent on the genotypic ratio. In case of like in Mendelian cross also we have seen this. So phenotype, what we see, phenotypic ratio, how it is dependent on genotypic ratio. So here you can see that first one, it shows the dominant character, like the visible character that we see in pea plant is the dominant one, right? So this is also dominant and this is also dominant, right? So 3 are the one which shows the dominant trait and 1 is the one which shows the recessive trait. So from the genotype, only the phenotypic ratio is being like defined. So the phenotype is dependent on the genotype of an organism, right? So it says that the genes provide only the potential for developing a particular phenotype, while the extent to which that potential is realized, it depends on the environmental influences and the random developmental errors. So from the diagram here, you can see that it is showing that the genotype, which is the genetic constitution of an organism, and the environmental influences and the random developmental events, both of these, they decide the extent to which a phenotypic character or the trait of an organism is visible, right? The expression of the physical trait. Phenotype is decided by these 2 factors, not genotype alone. Then coming to the Mendel's experimental design. So Mendel performed his experiment on the pea plant, right? So there was many advantages of using this pea plant. So pea plant usually it does the cross fertilization. The self-fertilization. It is a self-fertilizing plant. But Mendel tried to prevent the self-fertilization and tried to do the cross-fertilization in pea plants of the same variety and found out like certain conclusions he made out. So we'll be coming across those conclusions. So firstly, what did he do to prevent the self-fertilization in pea plant? Firstly, he removed the stamen from a developing flower bud before their anthers produced any mature pollen. So this is the pea plant, phenotype 1, it is showing, and phenotype 2. So both are having the stamen and pistil. So what did he do? He removed the stamens from a developing flowered bud. So from here, he removed the stamens. These stamens were being removed before the anthers were being produced. Any mature pollen would be produced in that particular this particular plant. So what did he do? He removed these stamens. After that, what he did was, wait a second. After that, what he did, the pollen from the stamens of the another flower. So from here, the pollens were being taken from another plant and it was being dusted onto the pistil. This is the pistil of the emasculated one to pollinate it, right? So from the plant from where the stamen was being removed, it is known as the emasculated plant, right? So in this plant, the pistil of this plant, these anthers were being dusted on this, and the cross fertilization was being done, and it was being seen that a pod was being developed, and these were the phenotypes which were being observed. So this is how he performed his experiment. Now what does he concluded from his experiments? Now Mendel worked only with the true breeding strains of the pea plant. What do you mean by the true breeding strains? True breeding strains means they will be present in their pure line. So the parents would be either homozygous dominant, or they will be homozygous recessive. No in-between strain would be present. Either it would be capital T, that is homozygous dominant trait for the tall, and it would be homozygous recessive for the tall trait, right? So homozygous traits he took. This is known as the true breeding strains. Now, he observed, Mendel selected 7 pairs of the traits to study in breeding experiments, right? And then he what did he do? Each pair affected one characteristic of the plant. So each pair which he took, it affected only one character of the plant. So tall one, it basically controlled this genotype, basically controlled the height, and the color basically controlled the seed color. So there were different genotype which control only one particular trait, not two traits were being controlled, right? Gene interaction is not taking place here. Only one genotype is responsible for one character. Then each member of a pair being clearly distinguishable. So each member of the pair, it was clearly distinguishable. So if it would be tall, it would be like, if it would be present in this form also, in heterozygous form also, then also this particular plant would be tall, right? And the other one was dwarf. There was only two conditions which were there, right? Not in between condition. There was no blending which took place. In incomplete dominance, what we study is, in case of Mirabilis jalapa, what happens? There is like two homozygous or two true breeding strains of two different plants as we take. Like one is producing the pink flowers, another is, one is producing the red flower and another is producing the white flower. Then when we cross these two, then what happens in F2 progeny, what we observe three different traits. The flower having the red color, the flower having the pink color, and the flower having the white color. So here the blending has taken place. But in case of like Mendel, the experiment which Mendel performed, there was no blending which took place. Like all the characters, they were distinguished from each other, and only like two of the possible outcomes were there. Right? So it showed the complete dominance in case of like whatever Mendel experiment he performed. Then the seven character pairs that he took while he was performing his experiment on the pea plant were, first one was the seed coat color, that is the flower color, and second one was the seed color. It was either yellow or either green. The pod color, either green or yellow. The pod shape, either inflated or pinched. Then comes the stem height. It was either tall or short. So you can see that all the characters, they are distinguished from each other, right? They are not something which is coming in between, either this or either that. Then comes the flower position. Either it was axial or it was terminal, right? Then the findings of Mendel. So he determined there are distinct units of inheritance, right? Which he called as factors. At that time, gene term was not being coined, right? So he called these units of inheritance as factors. Later, it was being the factors were being given the name as genes. Then he said the behavior of units could be predicted during the formation of gametes. So behavior of these units could be predicted during the formation of gametes. So when the gametes were being formed, they only decide what would be the behavior of a particular individual, right? Later researchers linked the behavior of the chromosomes during the meiosis to the Mendel's principle of inheritance. So whatever principle of inheritance that Mendel found, like there were three principles of inheritance or laws of Mendel. First was the law of dominance, law of segregation, law of independent assortment. So all these three principle of inheritance, they were being linked to chromosomal inheritance later on by the scientists, right? Then the study of the transfer of inheritance in this manner to offspring is called the Mendelian or the transmission genetics. So this is what we are studying about, the transmission genetics, which was being proposed by Mendel. So therefore Mendel is known as the father of genetics. Now here is a quick reminder for all of you. Now Shorter batch is being starting from 4th of March, which is not very far away. And here we'll be providing with you all the kind of facilities, such as the RAID classes would be there by CSIR NET experts. Then interactive online classes would be there, 24/7 doubt solving via chats. Printed hard copy would be provided to all of you, and video backups would be provided. Now, if you want to know more about the Shorter batch, you can call us into this toll-free number that is flashing on your screen, or either you can mail us, right? So the admission department, they'll guide you how to proceed. Then coming to the comparison between the homozygous and the heterozygous genotype. Now, what do you understand by the homozygous and heterozygous genotypes? So here it says that the homozygous is a genetic condition where an individual inherits the same alleles of a gene from both the parents, right? So here is, wait a second. So here is a parent, right? This is male, this is female. So from one parent, this particular trait, and from the second parent also this particular trait. This is being transferred to the offspring, and it is producing the homozygous dominant trait. So similar genes are being like they are being inherited to the offsprings, producing the genotype, right? This would be the homozygous dominance, or it could be homozygous recessive, right? So here it says the homozygous is a genetic condition where an individual inherits the same alleles of a gene from both the parents, right? So same allele it is inheriting. Second, it says the homozygous genotypes are represented as capital A, or A or small a for homozygous dominant or homozygous recessive condition respectively, right? Two different phenotypes are possible with a dominant or recessive homozygous conditions. So two different phenotypes are possible from homozygous dominant, homozygous recessive traits. Now, what would be these two phenotypes? So suppose if we take about the homozygous dominant for the tall character and homozygous recessive for the dwarf character. So two particular phenotypes would be there. Either it would be tall or either it would be dwarf, right? So two possible phenotypes could be produced from the homozygous dominant or homozygous recessive genotype. Then it says that the homozygous genotype results in a single type of gamete. Only a single type of gamete would be produced. Homozygous genotype produce the same traits over different generations, right? They like during the generation to generation, this homozygous genotype, it doesn't change. It says that it produces the same traits over different generations, right? So variability in case of like organisms, it is not due to homozygous alleles. It is basically due to the heterozygous one. Due to heterozygous one only, the variability in the different organisms is being found. So heterozygous character is more important as compared to the homozygous one. Then it says that the homozygous condition doesn't show hybrid vigor. So it doesn't show hybrids here, right? Because like from both the parents, that alleles that is being like inherited to the offsprings, it is coming like the alleles are same. Therefore, there won't be any hybrid which would be formed, right? There won't be any crossing over which will take place. So the homozygous dominant and the homozygous recessive are the two types of homozygous conditions, right? So this is all the same. The heterozygous, for heterozygous, it says the heterozygous is a genetic condition where an individual inherits different alleles of a gene from two different parents. So for this, the similar example we can take here is like this is one parent, this is the second parental chromosome. So here is the capital T, this is the small t. So two different allelic form of a gene. Now these are being inherited in case of offspring, where what happens, both of these like these genes are being inherited and it forms a particular genotype. And this is called the heterozygous genotype, right? This is the heterozygous form. And these two alleles are different from each other. One is representing for the tall and other is representing for dwarf, right? Heterozygous genotypes are represented by capital A, small a genotype. So this is here it is saying. And then it says that the phenotype is mostly due to the dominant allele in the heterozygous condition. So dominant allele, it basically masks the expression of the recessive allele, and only dominant one is visible in case of like when this offspring is, the genotype is present in heterozygous form, right? Then it says the heterozygous genotypes produce different traits over different generations. So different traits are produced in different generations, and there is a variability which is being is being shown, and hybrids are being produced in case of heterozygous. Then it says that the heterozygous condition can be expressed in three different ways. These heterozygous condition can be expressed in three different ways. It could be codominance, which we see in case of blood groups. So in case of blood groups, both A blood group and B blood group people, that is the AB blood group people are there, where blood group A and B both are present in them. In case of incomplete dominance, there also this heterozygous condition is expressed. Taking the example of Mirabilis jalapa, where there is a red plant, like a red colored flower is there, then there is a pink colored flower is there, and then there is white colored flower is there, right? So incomplete dominance took place here, blending took place, like completely this red color homozygous trait didn't express itself, right? Then comes the complete dominance. Incomplete dominance, like either it would be tall or it could be dwarf. So this is the major difference between both homozygous and heterozygous genotype, right? From the figure, we can see that the dominant allele and recessive allele, two different types of alleles are given. So genotype is being given capital S, which is the homozygous form. This is the true, true trait, or the pure trait, you can say, right? This is the homozygous dominant one. This is the heterozygous one, and this is the homozygous recessive form. So the homozygous, both like homozygous dominant and heterozygous dominant, both of these, the phenotype for both of these are same. Like the dominant one will mask the expression of the recessive allele. As a result, the phenotype which it will show would be for the dominant trait. So smooth seeds would be shown in both cases, like homozygous dominant and heterozygous dominant, and only wrinkle seeds would be shown only in case of homozygous recessive one. So this is the basic difference. Now coming to the monohybrid cross. Now, as we know that Mendel did his experiment on the two true breeding parents. Now here is the parental generation. It shows where smooth seeds are taken and wrinkle seeds are taken. So these would be heterozygous dominant, and it would be heterozygous recessive one, right? So smooth seed, the genotype would be capital S. For this, small s would be there, right? So in F1 generation, what happens? All smooth seeds are being produced, right? How? Now it will produce the allele that it will be producing would be capital S, and this would be producing small s. When these would be crossed, then it will be producing the heterozygous form, capital S and small s. So this capital S, it will dominate over the recessive allele. As a result, all the F1 generation would be, all smooth seeds would be there, right? So this is how it will take place. Then in this F1 generation, what happens after that? The selfing is being done for F1 generation. So F1 generation is being crossed with its similar genotype. So F1 is being crossed with F1, and what happens here? The selfing is being done, and the F2 generation is being produced, where half of you can see that how many 2.96 it's saying, like it's approximately 3. So 3 are showing same phenotype, that is the dominant one, that is the smooth, and the one is the one one is that which is showing the trait for the wrinkled one. So it is the recessive one, right? Now in F2 generation, what happened here? What did you observe? In F1 generation, we saw that the recessive trait, it was being suppressed, right? And F2 generation, again the recessive trait, it reappeared. Now this was some like magic which happened. Now coming to the another diagram, it says that it is like the Punnett square, it is being made, and the same experiment is being it is being explained with the help of Punnett square. Now here it says that the F1 generation for male and for female, it says that the both smooth seeds, both are having the smooth seeds. So in F1 phenotype, the result that was being observed, it was the heterozygous form of these seed, right? Capital S and small s. And this was the diploid F1 genotype. Now coming to the haploid F1 genotype. Now diploid would be produced in anaphase 1 and haploid would be produced in anaphase 2, right? When the chromosomes, the sister chromosomes, it will separate from each other towards the pole, and 4 different haploid would be produced here. So what happens? The haploid F1 gametes, one carries the capital S, second carries the small s, one carries the capital S and small s, right? This is the haploid F1 gamete formation which took place. And now what happens? These were being self-crossed. Now selfing was being done in F1 generation. Then what happens? F2 generation was being found, where it was being shown. So from these are the two one, the capital S, small s, capital S, small s. So from here, the one which would be coming, it would be having the half effect, and it would be having the half. This would also be half, and this would also be half, right? So what it says here is, yeah. In case of F2 generation, when the Punnett square was being made and crossing was being done between the two F1 gametes, the selfing, when it was being done, it was being found that the genotype ratio that was being found was 1/4 were homozygous dominant one, 1/2 were heterozygous dominant, 1/4 was homozygous recessive one. So the ratio was 1:2:1, right? And in case of F2 phenotypes, like the phenotypes which was being observed were 3/4 smooth seeds and 1/4 wrinkle seeds, right? So the phenotypic ratio was basically 3:1. So from the Punnett square itself, you can see like how this phenotypic ratio and genotypic ratio was being formed, right? So here is the recessive trait. So here is the recessive trait. Here are the 3 dominant ones, out of which 2 are showing the heterozygous dominant, that is capital S, small s. This is present in its heterozygous form, while 1 is present in its homozygous form, right? So ratio for the genotype was 1, this was 2, and this was 1. So 1:2:1 was the genotypic ratio. While coming to the phenotype, all these three, they were showing the same phenotype, that is the dominant one. So this was 3 and this was 1. So the ratio, phenotypic ratio, became 3:1. So it says that the monohybrid cross is basically the genetic cross involving a single pair of gene. That is, one trait is involved here. So in monohybrid cross for Mendel, here 2 laws are majorly applicable. First is the law of dominance, which you can see in case of when the two parental pure breed, they were being crossed with each other, and F1 generation, you found that the heterozygous genotype which was being formed. So there what happened, the dominant allele, it suppressed the expression of the recessive allele. So law of dominant played a role over there. So law of dominant, what it says, one allele masked another, one allele was dominant over the other in F1 generation that we found out, right? Then coming to the law of segregation. Law of segregation is also being applied here. Now, law of segregation is something which is a universal law, which you can say as the true law. None other law of dominance and law of independent assortment are also not the true law. Law of segregation is the one which is the true law, because meiosis will eventually take place, and when meiosis take place, law of segregation is being applied there. Now recessive traits which were being masked. Now here comes the factor where the recessive traits which were being masked in the F1 generation from the true breeding crosses, they reappeared in the specific proportion in case of F2 generation, right? Now you can see here that in specific proportion, the in F2 generation, these recessive traits were being reappearing, right? One was produced here. The recessive trait was being produced in F2 generation, which was being suppressed in case of F1 generation. Now, 2 members of a gene pair, alleles, which we call, segregate or separate from each other during the formation of gametes in meiosis. So in case of meiosis, what happens is, like how meiosis actually takes place. Let us look at it. So what happens here? So here is the parental chromosome, right? So it is having one allele for capital R. Here it is having small r, right? This is the parental form. Now what happens when replication take place? Then it will be having 2 of these alleles, both of these, right? For both male and female. Then what happens when prophase 1 takes place in case of meiosis? In prophase 1, then what happens? There would be crossing over which will take place. As a result, these homologous chromosomes, they'll cross with each other and they'll produce capital R and small r. And here it will be producing capital R, small r, right? So we can erase this. It's not required. Right. After that, what happens? After prophase 1, then comes anaphase 1. In anaphase 1, these 2 homologous chromosomes, they'll separate from each other towards the pole, right? So separation takes place. After anaphase 1, when anaphase 2 happens, then these two sister chromatids, they'll get separated, and 4 haploid cells would be produced consisting of capital R, small r, right? In capital R, small r. This would be the FN progeny in haploid form that we have studied, right? Haploid FN progeny. And this was the diploid FN progeny that we studied, right? So in anaphase 2, what happens? Anaphase 2, the haploid genotypes are being produced. So there where the segregation basically takes place, the chromosomes, they separate from each other, right? Two different alleles, they separate from each other. So that's what it is saying here. So it says that the two members of a gene pair, which is known as allele, segregate or separate from each other during the formation of the gametes in meiosis. So this is how in meiosis the separation takes place. So segregation is a universal law. It is a true law which will take place because meiosis has to take place, right? So law of segregation will occur. And then it says that each gamete, it carries only a single allele of each gene. So each gamete which will be producing, it will carry a single allele of each gene, right? Then the conclusion that was being made for monohybrid cross. So the first was the result of the reciprocal crosses were always the same, right? So like here, another factor came, the reciprocal cross. So reciprocal cross, when the results are same, what does it mean? Like reciprocal cross, if we know, it is a 2 hybrid cross, first of all. So let us take an example here. So this is a female. So sorry, this is a male, this is a female. So character here we have taken is capital T, small t, small t. And here we take another, and here is a male which produces small t, and here is a female which produces capital T, right? Now in FN generation, capital T, small t is produced. Here also in FN generation, capital T and small t is produced, right? In both the cases, the offsprings, they're having the same genotype, right? The genotype remains same. That is, it is not dependent on the source of the gene from either of the parent. So here it says that it is the complete dominance which is taking place. And the result of, or the expression of the genotype in case of offsprings, it is independent of the parental origin, right? So results is same in both the cases. Either it comes from like male or female. Like in this case, the male was like homozygous dominant one. Here, the male is homozygous recessive. Then also the trait in case of offspring, it remains same in both the cases, right? This is known as the reciprocal cross. It is a 2 hybrid cross, and it basically tells that it is a nuclear division which is taking place, not the chromosomal one. So the result of the reciprocal crosses were always the same, right? First conclusion that he made. Secondly, it was being seen that all the FN progeny resembled one of the parental strains, indicating that the dominance of one of the allele over the other, right? So FN progeny, it resembled one of the parent. And the parent that it resembled was the dominant one, the homozygous dominant one, right? If the two like traits are two pure breeding traits are being taken from the parents, one is the homozygous dominant for tall, and one is the homozygous recessive for tall character, and both of them, when they are being crossed in FN progeny, all progenies that are produced, they are all tall, which means that the dominant one, the dominant allele, it suppressed or it masked the expression of the recessive allele. So second conclusion, this was what was being made. Third was in F2 generation, the parental trait that had disappeared in case of FN generation, it reappeared. Here the law of segregation basically takes place, right? Because meiosis took place, and as a result, when the haploid, like when the selfing was being done in FN generation, then the F2 generation, they had the recessive traits reappeared in them. So law of segregation took place here, right? Here law of dominance was playing the major role, right? Then comes that the trait seen in the F1, that is the dominant trait, was always found in the F2 at about 3 times the frequency of the other trait, right? So the trait which was being seen in F1, the F1 with the dominant trait which was being seen in F1, it was being visible in F2 generation 3 times the frequency of the recessive trait. So the ratio here became 3:1. For phenotypic ratio, it became 3:1, right? So the genotypic ratio was 1:2:1. We have already seen how 1:2:1 came, right? Selfing of the F1 generation took place, and in the F2 generation, after the selfing of F1 generation, the genotypic ratio it came was 1:2:1. And for the phenotypic ratio, it came 3:1, which means there is the complete dominance which is taking place, and there is no blending inheritance which takes place. These are the two important terms, because we study like there are how many types of dominances are there. Like there is codominance is also there, incomplete dominance is also there, and complete dominance is also there. So in question, it will always be mentioned which kind of dominance which type of dominance it is basically talking about. If it is talking about the complete dominance, then the ratio would be 3:1 always. And sometimes it is not 3:1, because when there is a lethal condition observed. So lethal condition, this is something lethal allele which takes place. That is some other concept. We'll be discussing about it like some other time. But in that case only, what happens in case of complete dominance, the ratio is not 3:1. The ratio is 2:1 at that particular point of time, right? And in case of so there is no blending which is taking place. Another case of dominance is the incomplete dominance, where what happens in case of Mirabilis jalapa, like there is one a red type is being produced, right? Two pink type is being produced, and then one white colored flower is being produced. So this is kind of blending is taking place here. That it is not a complete dominance taking place, right? So phenotypic ratio for complete dominance is always 3:1. Exception is there, the lethal allele case, and there is no blending inheritance which is taking place. So we can say that this is condition when the ratio is 3:1. It is the condition of complete dominance, right? So this much are you all clear with? Yes, incomplete dominance. Kiran Kumar, she says that the incomplete dominance for that, the ratio is 1:2:1. The phenotypic ratio is 1:2:1, and the genotypic ratio is also 1:2:1, right? Could you please repeat? What do you want me to repeat, Mukesh? Where have you got stuck? Okay, Mukesh, you came late now. So this video would be there only. Like you can go back and check again, and then you'll be able to understand, right? So here is the branch diagram for the monohybrid cross that is being shown. Now in question paper, what happens when you have to calculate, do the calculations and take out the probability and all that stuff? You cannot like always do the Punnett square stuff. It takes a lot of time. So at that particular point of time, what you can do, you can make use of these branch diagrams. These are very much helpful. So here it says that the F1 generation, as we know, in case of F1 generation, the genotype which is being produced is capital S, small s, right? The selfing is taking place here. So this is the selfing which is going on, right? And the gametes that are being produced, half of them are capital S, half of them are small s in both the cases, right? Then what happens? Random combination of gametes results in. Now when the selfing is being done in F2 generation, what do we observe? This is the like one of the allele for one of the parent. So half of capital S and half of small s is being taken here, right? Two different chromosomes, they are present. Now this half capital S, when it when it combines with the other parents half capital S, then what is the ratio that is being produced in F2 progeny? It is like 1/4 ratio is being produced, and the trait which is being shown, the genotype which is being shown is capital S, right? So 1/4 capital S being formed here. And in case of when this half capital S of one parent, it combines with the half small s of the other parent, then what it produces? It produces 1/4 capital S, small s, right? So this is the genotype which is being produced. Heterozygous genotype is being produced here in 1/4 ratio. Then coming to the other chromosome having of the other, like one of the parent having the other chromosome, which is having the allele that is for the recessive trait, right? Single allele for the recessive trait. So when this combines with the half capital S of other parent, then it produces 1/4 ratio of the heterozygous genotype, right? Both these heterozygous genotype from both the chromosomes, when they combine to each other, they produces a ratio of 1:2. This is basically the genotypic ratio I'm talking about, right? And when this half small s of one parent, it combines with the half small s of the other parent, then it produces a 1/4 ratio of the genotype, recessive genotype, right? Small s, small s. So what you can see is one of it is in homozygous dominant form, two of them is heterozygous dominant form, and one is in homozygous recessive form. So genotypic ratio which came here is 1:2:1, right? This is through the branch diagram you can see. Now for coming for the phenotypic ratio. Now how can you calculate the phenotypic ratio here? Now for F2 progeny, the phenotype is being given here. 3/4 capital S is being formed, right? And here dash is being given, which could be like it could be capital S or small s, right? Like this is how it is being given. 3/4 capital S, or it could be either S, capital S, or it could be either small s, right? In both the cases, the dominant character would be visible. And in case of recessive one, it is being showing 1/4 ratio. That is small s, small s. So this would be only one which would be produced, and this would be three. Three how? Like one would be capital S, and other would be capital S, small s, and these would be two in number, right? So three are being in dominant state and one is in recessive state. So 3:1 is the phenotypic ratio. This is how through branch diagram you can calculate the genotypic and phenotypic ratio. This is for the monohybrid cross, like for dihybrid cross, and for trihybrid cross also you can calculate it like that. Kumar Mukesh says that how can we relate incomplete dominance with the codominance? Now, incomplete dominance is basically what it says, that there is not the trait is not completely visible in the F2 generation, right? There is a middle thing which is being produced, that is the blending, it takes place there. In case of in case of codominance, what happens? Two of the different traits, they are visible in the in the offsprings, which means there is no blending. Both of these traits are different from each other and they're visible in the progeny, but the blending doesn't take place here, at that particular point of time. In case of incomplete dominance, blending takes place, right? How to remember this ratio if characters are more? Yeah, so we'll be discussing about that also. So firstly is monohybrid cross, if you are clear with it. Dihybrid cross and trihybrid cross, you can take care of it. So crosses I'll be discussing here. There are different types of crosses. Now selfing, if you would have seen here, what did you saw? In selfing, you saw that for phenotype, right? For the dominant case, this I'm telling you about the selfing, the crossing, the selfing. In case of selfing, what happens is for the phenotype, for the dominant trait, the ratio is always 3/4, right? And for the recessive trait, the ratio is always 1/4. This ratio you need to remember in case of questions solving also. If you remember what kind of cross is basically taking place, whether it is selfing taking place, whether it is test cross taking place, whether it is outcross taking place, or reciprocal cross which is taking place, you should know like what ratio would come for the dominant and the recessive trait. For selfing, for the dominant trait, the ratio which comes is 3/4, and for recessive trait it comes is 1/4. Then comes to the test cross. Now, what do you mean by the test cross? In case of test cross, what do? We cross one of the parental, one of the parent produced in F1 generation. It is being crossed with the recessive, one of the recessive parent, right? So this test cross is majorly being used to know what is the genotype of the parent, the dominant parent. What is the genotype? It could be either heterozygous form, right? It could be heterozygous parent, or it could be a homozygous parent. So what is the type of dominance in the parental genotype for understanding that, of to determine that test cross is being done, right? So the test cross is being given here where it is being seen that the genotype of the parent we don't know. This is the F1 generation, right? The genotype of the parent we don't know, but it is the dominant trait that we can say, but it is heterozygous or homozygous that we don't know. And then it is being crossed with one of the like homozygous recessive parent it is being crossed with. This is the homozygous recessive one, right? Now what happens when we draw the Punnett square? Gametes from the recessive parent, it is being given here, and gametes from the parent of the unknown genotype, the genotype that we don't know. Now what happens is the ratio that is being given here. So here we can see that all are the test cross resulting in all dominant offspring. So all are produced in dominant state, which means that the genotype, right? The genotype of the parent, it would be capital Y, capital Y. Then only it can happen that all the progeny, it would be in their dominant form, right? The dominant trait it would be showing, right? So it would be homozygous dominant form. Then only from this we can infer that from this test cross result we can infer that the genotype of the parent is homozygous dominant one. For the homozygous, for the heterozygous dominant, what happens is, here is the result of the Punnett square of the heterozygous dominant. So here we can see that half of them are recessive trait and half of them are dominant. So half is dominant and half is recessive. The ratio here comes is 1:1, which means the phenotypic ratio here comes is 1:1, which means that the parent is in heterozygous genotype. The parent is consisting the heterozygous genotype. So it would be present in capital Y, small y form, and here it would be present in capital Y form, right? This much everyone is clear with. Is it necessary to have homozygous dominance? Oh, I didn't understand. Like, it is necessary for what? I'm telling you about the two conditions here. It could be homozygous dominant or it could be heterozygous dominant, which could take place. Through test cross, we basically determine what is basically the genotype of the parent, the unknown parent, right? And coming to the second type of cross, that is the reciprocal cross. So reciprocal cross that I have told you earlier, that it is kind of the two hybrid crosses here takes place, right? The two types of crosses here is taking place. One is this and another is that one. Wait a second. Yeah. So two hybrid crosses here takes place. Now, here you can see that the female one here is dextral coiling is taking place. Here synestal coiling is taking place in case of male, and here in case of it is being reversed. So the genotype of the parent is being reversed in reciprocal cross. And for both of these crosses, what we observe that the offspring, the result, it is same. The results that is being produced in both of these crosses is same, right? If we change the genotype of the parent. But the conclusion that is being made here, it is a kind of firstly, the result or the expression of the phenotype, the expression of the offspring, the expression of the genotype of the offspring, it is not dependent on the parental origin, right? It is not dependent whether the male is showing the dextral coiling or the female is showing the dextral coiling. The result for the offspring, it will remain same. So first infer is this. Secondly, it basically says it is the nuclear inheritance is basically taking place here, not the mitochondrial inheritance. In case of mitochondrial inheritance, it is dependent upon the result or the offspring's the genotype expression of the offspring. It is dependent upon one of the parents' genotype, right? And then it says it is a uniparental, like it is a biparental effect it is being showing, because both the parents, the expression of the genotypes of both the parents are responsible for the expression of the genotype in case of the offsprings, right? These are three kind of infer that you can the result that you can produce from the reciprocal cross. So one is the test cross, another is the reciprocal cross, and another is also there, that is the outcross. In outcross, what happens? The F1 generation progeny, it is being crossed with the homozygous dominant parent, right? There what happens, all the offsprings which are being produced, they are all dominant, right? So 100% dominancy is being shown at that particular point of time in case of outcross. So there are three types of crosses here. Test cross, I have discussed. Reciprocal cross is there, and then there is outcross. So in case of test cross, what you have seen here, the ratio that you can see is 1:1 ratio that you have seen here. For the recessive, for the dominant, it is being for the recessive. Sorry, this is the recessive one. For the recessive, the ratio is 1:2, and for the dominant also, the ratio is 1:2. Right. So there are two crosses where basically the segregation takes place. One is the test cross, and another is the selfing, where the segregation of the chromosomes basically takes place, right? So the ratios in case of selfing and the ratios in case of test cross, you need to remember for both dominant, like dominant allele and recessive allele. Then only you can understand like how to solve a particular question. So here is the two questions for all of you. So whatever we discussed till now, these questions need to be solved through that observation through them only. So first it says that the Mendel crossed the tall pea plants with the dwarf ones, right? The FN plants were all tall. When these FN plants were being self to produce, the selfing is being done and the F2 generation was being formed. Then it says that he got a ratio of 3:1. So tall to dwarf ratio is being in the offspring was being produced. Then it says that what is the probability that out of the 3 plants of F2 generation picked up at random, 2 would be dwarf and 1 would be tall, right? So what does it says? The FN plants were all tall. So FN progeny would be all tall and it would be in heterozygous form, right? It would be it would be capital T, small t. This would be the genotypic ratio. Let us suppose we are taking for the trait that is the plant height. So FN progeny is this. Now it says that when these FN plants were being selfed, so selfing was being done here, right? Selfing was being done and it produces the F2 progeny. In F2 progeny, it was being seen that a 3:1 ratio was being found. Capital T, small t, these were 2 which were being produced. This were 1 and small t, it was being produced 1. So 3 were all tall and 1 was the recessive one. Then it says that what is the probability that out of these 3 plants, now out of these 3 tall plants, what is the probability? No, out of these like 4 plants, now what is the probability that out of 3 plants, so 3 plants we took at random, we took any 3 plants of the F2 generation picked up at random, 2 would be dwarf and 1 would be tall. So 2 should be dwarf, right? And 1 would be tall. So for this type of questions where there is a binomial theorem, like a binomial thing is taking place, 2 conditions is being given. In this case of, in these kind of questions, you need to apply the binomial theorem here. So what does the binomial theorem actually says? So binomial theorem is basically P plus Q to the whole power n. It could be anything, right? Now, here n is basically 3 is being given here, right? Because 3 plants we are choosing. So what happens when we, like the bracket when it is being open, then it will be P cube plus Q cube would be there. Then there will be 3P square Q plus 3PQ square. This would be formula for the when n is equal to 3, right? When n is equal to 2, then what happens? It would be P square plus Q square plus 2PQ, right? Now here it is being given for the 3:1. Now it is being saying, let us consider that the P is the dominant for the dominant trait and Q is for the recessive trait, right? If this happens, if this condition we have taken, then what happens? It is saying that the 3 plants are being taken, right? Out of which we need to say, we need to pick that 2 would be dwarf, that like Q square would be there, and 1 would be dominant one, right? So for this, what happens? Q square and there would be P and there would be 3 which will come here. If we say about like P plus Q to the power n, where n is basically 3. So when we'll open this one of the binomial expansion would be 3PQ square, right? So here it is saying about dwarf. Now here it is saying that selfing is being done. And I told you about selfing was that for the dominant trait, for the selfing, what happens? The ratio is 3:4 for the dominant trait. And for a recessive trait, in case of selfing, the ratio is 1:4, right? So you can apply for Q this particular ratio and for P this particular ratio. Then the result which we will be getting is for the P it would be 3/4 and for Q it would be 1/4 to the power square. Now all of you can, I know, like you will be able to solve this and tell me what would be the answer for this. Okay, Keithna says it would be 3/8. Amit says it would be 3/4. Sunny Sharma. Don't make it more complicated using the chi-square test and all. Binomial expansion is enough here, right? So firstly, you should know like how binomial expansion basically like how the bracket gets open, how different kind of binomial expansion basically takes place. After that, only you can infer which where to put like which is the dominant trait, what would be the recessive trait, and what is the ratio for the selfing. And if here it would be given test cross, the ratio would be completely different at that particular point of time, right? So yeah, so the result here would be coming 9/64. This would be the result, right? This ratio would be there where 2 would be dwarf and 1 would be dominant when we are taking 3 plants in random, right? So I hope everyone would have understood this. No, the option is C. The correct option is C, 9/64. So Amit Kumar, yeah, you were right. Ankita also right. Kiran Kumar, yeah, you are also right. Yes. So C is the correct option here. Now for the second question, it says that the genes A, B, and C control 3 phenotypes, right? Genes are being given A, B, and C, and now they control the 3 phenotypes which assort independently, right? Independent assortment, we'll be studying about what is independent assortment. This law of Mendel, it basically comes in dihybrid cross, right? A plant with the genotype capital A, small a, capital B, small b, capital C, small c is being selfed. So here also selfing is taking place, right? Now this is the genotype which is being given here, small b, capital C, small c. Now selfing is being taking place here, right? So for selfing, we know what is the ratio. For dominant one, it is 3/4, and for recessive one, it is 1/4. What is the probability for the progeny which shows a dominant phenotype for at least one of the phenotypes controlled by genes A, B, and C, right? Now, for this question will be coming after we study about the dihybrid cross, because independent assortment also comes there, and the ratio that will be coming across here, it is basically regarding the dihybrid cross. So first we will be studying about dihybrid cross, then we'll be coming to this question, right? So save this question for later. So coming to dihybrid cross. So in dihybrid cross, what happens? We basically takes 2 different traits, right? 2 different traits are being taken here. In case of monohybrid cross, we were taking just one single trait and crossing was being done. So here what happens in F1 generation, both male and female. Now in F1 phenotype, so firstly we'll be going through this particular diagram. So here is the parent, female parent, here is the male parent. Now the parental phenotype, which is in homozygous form, homozygous recessive and homozygous dominance, pure breed. So here 2 traits are being given here. That is for the smooth and yellow, the seed color and the seed shape. So for seed shape, for seed shape, the genotype is capital S for one of the parent, that is homozygous dominant, and for seed color, it is capital Y, that is homozygous dominant, right? For here also, it is homozygous recessive for the other parent. So both are in true breeding form. Now here it says that the haploid gametes which are being produced, it would be having, so here from here capital S would be coming, from here capital Y would be coming, right? And from here small s would be coming, and from here small y would be coming. Now it says that in F1 generation, when the crossing is being done, then what happens? The progeny which is being produced, it is capital S, small s, capital Y, small y. This is the progeny which is being produced. This is the heterozygous dominant form for both of the characters. That is the smooth plus the yellow colored seed would be produced, right? So this is the F1 generation, the progeny which is being produced, right? Now it's when in F1, after the production, the F1 generation is being formed, then we'll be doing the selfing of this F1 generation and F2 progeny would be produced. Now, how many genotypes would be produced here? How many genes would be produced here? For that, there is a simple formula, 2 to the power n. Since where n basically denotes the traits, number of traits that we have taken. Since here we have taken 2 traits, one is the seed color, one is the seed shape. So we'll be doing 2 square. So there would be 4 different genotypes or gametes which would be introduced, right? So when we do selfing, here is the diploid F1 genotype and here is the haploid F1 genotype. When the selfing is being done, you can see that 4 different gametes are being produced. One is the capital S, capital Y, dominant one, capital S, small y, small s, capital Y, small s, small y. Now, how it is basically being produced? Now, if we suppose we have taken here capital S and small s, right? This is the chromosome, 2 different chromosomes, and this is from one of the parent, right? Now, this can combine with either capital Y or small y, or it can combine with either capital Y or small y. Then the progeny which would be produced here, it would be capital S, capital Y. For this, it would be capital S, small y. For this, it would be small s, capital Y. And for this, it would be small s, small y. So 4 of the different genes which are being produced here, right? This is how the genes are being produced. So when in haploid F1 gametes, these are being selfed with each other, then what happens? The phenotypic and genotypic ratio at last which is being observed. Now this through the help of Punnett square we have done here, selfing is being done. So we can see that one is produced here which is homozygous recessive one. This is the one which is homozygous dominant one, right? Rest all one, it lies in between. Only 2 are the one which are having the pure traits which are coming directly from the parents. Now the ratio here, what happens is, for F2 genotypes and for F2 phenotypes. Now genotype, what happens? 1 by 16, right? 16 genotypes are being produced here. Out of those 16, only 1 would be having the homozygous dominant trait, right? Out of 16, 2 would be having capital S, small s, and capital Y form, right? And 2 by 16, it would be having capital S, small s, and capital Y, small y. So here it is being given genotypic ratio, right? So genotypic ratio, how can we calculate through monohybrid cross? That is the major question, right? Now in monohybrid cross, what happens? The genotypic ratio is basically 1:2:1, right? So in case of dihybrid cross, it would be square of this ratio. So when we multiply this 1:2:1, then the genotype which will be coming would be 1:2. This would be multiplied by this 2. It would be 2:2, 1:2, then 2:2, 4:2, 2:2, 1:2, 2:1. This would be the ratio. So multiplication, all you have to do, if you remember the monohybrid genotypic ratio, you can calculate for dihybrid, even for the trihybrid also, right? For the phenotype also, it is the same. 3:1 is there. You just do the squaring of this, right? So multiplied by 3:1. So the ratio here, phenotypic ratio, which will basically coming here would be 9:3:3:1, right? Out of which 9 would be the dominant trait, 3 and these would be basically, 1 would be the recessive one and this would be the intermediate one, both of these 3s, right? So here it is saying the result is being given here. For the genotypic ratio, it is being given here, and for that the phenotypic ratio, it is being given that 9 out of these 64 which is being produced, it would be smooth and 16 which is being produced, it would be smooth and yellow seeds, right? This would be the dominant trait. 3 would be smooth and green. Like one trait would be in the dominant form and the other would be in recessive form. This would be the 3 one, and for the second third which comes is basically for the wrinkled and the yellow seeds. One of the trait would be in dominant and other would be in recessive form. And last one which is left, like one only is present here, the homozygous recessive form, which would be all for both the traits, it would be in recessive form, right? So this would be more clear when we do the branch diagram that we did for the case of the monohybrid cross, right? Now, firstly, let us study about the conclusion of the dihybrid cross. The conclusion for the dihybrid cross, it was the F1 are the homozygous for the two pairs of the alleles at two different loci, right? Such individuals are called the dihybrids. So for the dihybrid cross, basically we took the two traits and we crossed each other. This is the basic definition of the dihybrid cross. And a cross between the two of these dihybrids of the same type is called the dihybrid cross. Now, the basic law of Mendel, which comes in case of dihybrid cross, is the law of independent assortment. So what does the law of independent assortment says? It says that the pairs of the alleles of the genes on different chromosomes segregate independently in the formation of gametes. So when the gamete is being formed, like here I have explained here, when the gamete was being formed, the gamete from one parent, we took capital S and small s. So these two different gametes, it can independently assort with any of the gamete of the other trait, right? It could be capital Y or small y, right? Independently, it can assort with any one of them, and it can produce four different possibilities are being produced here. 4 different gene possibilities are being produced here. It is not that at a particular point of time all these 4 would be produced. That is the possibility which would be there. So pairs of the alleles for the genes on different chromosomes, they segregate independently. Firstly, they will segregate independently and in the formation of the gametes. Right. Then it says that for the principle of the independent assortment in a dihybrid cross, the F2 phenotypic ratio is 9:3:3:1 for the 4 phenotypes. Right. So the 4 different phenotypes would be produced here. 2 to the power n is the formula through which we calculated that. Now here is the branch diagram through which we can calculate the genotypic and the phenotypic ratio in a better way for the dihybrid cross. So here it says the selfing is being done for the F1 progeny, right? So in F1 progeny, the heterozygous genotype which was being produced was smooth and yellow, and this was basically being crossed with each other. And in F2 progeny, what happens is, so 3:4 ratio it is being taken here, right? 3:4 ratio, from where does it came? It came basically from the monohybrid cross. In case of monohybrid cross, what did we saw? For a dominant trait, the ratio is basically 3:1. So 3 out of 4 possibilities, 3 out of them are in dominant state. So 3:4 is being taken here, and then it is being taken capital S, and this is the this dash is being given, fill in the blanks, which basically says it could be capital S or it could be small s, right? Any of the two alleles it could be. And then it is being crossed with 3:4, that is the dominant phenotype of the other trait, that is capital Y, and dash is being given. It could be either capital Y or it could be small y. Then the ratio which would be produced, it would be 9:16, right? 3 into 3, 3 3 is 9, and 4 4 is 16. So 9:16 ratio would be produced for capital S dash and capital Y dash. This dash could be either it could be capital S small s, or this could be capital Y or small y. Then coming to the other one, and this allele of one parent in ratio of 3:4, it can also independently assort with other allele of the different parent of the other parent, and it would be in the ratio of 1:4. That is the green phenotype which it would be showing, and the genotype it would be small y small y. That is the homozygous genotype would be there. So for that, what happens? 3:4 multiplied by 1:4, the ratio would be 3:16 here, right? This dash could be capital S or small s. Any of the allele could segregate with or assort with this particular homozygous recessive form of gene, right? So here the phenotype which would be produced, it would be smooth and green. Like one of the trait would be dominant form and another would be in recessive form. And in earlier one, both of the traits were in dominant form. Then coming to the wrinkled one, other. So this would be present in one chromosome, this would be present in other chromosome, right? Two different alleles for one particular trait. So this is smallest. It is representing smallest, which would be present in 1/4 ratio, as we have seen in case of monohybrid cross, right? In monohybrid cross, the recessive trait, it was having one, like only one was there, which was present in homozygous recessive form. So 1:4 is the ratio for this particular trait. Then what happens? It can independently assort either with the dominant one or either with the recessive one, right? For the dominant, it could be either present in the homozygous form or it could be present in the recessive form. Then the ratio that would be there, it would be 3:16, right? 1/4 multiplied by 3/4, it would be 3/16. Out of which one of the trait, it would be in dominant form, other would be in the recessive form. And when this particular homozygous recessive form of one parent, it independently assorts with homozygous recessive of the other parent for the different trait, then what happens? Both of the traits, they are present in recessive form. This is the homozygous recessive form, right? So the ratio here comes 1/16. So the phenotypic ratio which came for the dihybrid cross is 9:3:3:1. This I have explained with the help of branch diagram, which was like much better as compared to the Punnett square that we are making. It would take a lot of time. So did everyone understood till here? Okay, now we'd be coming to this particular question. Right. So the question says, like, you have already read the question, and for the B is the correct one. Okay. So Kumar Mukesh says B is the correct one for the second question. How did you calculated this? Now, here what is it is saying, the last line, if you read it, it says that what is the probability for the progeny which shows the dominant phenotype for at least one of the phenotypes, right? At least one of the phenotype, either A, B, or C, it would be present in dominant form, right? Or all of them can present in the dominant form. This could also be the condition, right? So when we were doing the dihybrid cross, this is basically related to trihybrid cross, right? So either it is trihybrid cross or dihybrid cross or monohybrid cross. What we see is, for the phenotypic ratio, in case of monohybrid cross, what did we see? It was 3:1 ratio, where the three individuals, they were in dominant form and one was in recessive form. Now calculating for the this trihybrid cross, what we can do the cube of this particular ratio, this phenotypic ratio. What would be the answer for the cube if we do? Can anybody tell me what would be the phenotypic ratio for trihybrid cross? This is the way how we can calculate the phenotypic ratio. Yes, Mukesh, I'll be going slow. Firstly, I want you to calculate this cube of this ratio for the phenotypic ratio. 3:1 cube. What would be the answer for this? Anybody? Now you can also split it. Like how? In case of dihybrid cross, what we saw, for the dihybrid cross, the phenotypic ratio was 9:3:3:1, right? And this was for dihybrid cross, and for monohybrid cross, it was 3:1. Now when we multiply this, then what happens? 27:9:9:3, right? 9:3:3:1. This is the phenotypic ratio for the trihybrid cross which came. Was everybody able to understand this? Yes, this is the ratio. Now here what you can see is only one trait, right? Let me write it again. So the ratio here basically is 27:9:9:3:9:3:3:1. 3:1. This is the phenotypic ratio for the trihybrid cross, right? Here you can see there is only one trait which is in recessive form. This would be the recessive one. This would be in complete dominance 1. 27 would be in complete dominance, right? And these would be lying somewhere in between. So here the question says that at least one should be dominant. Means in these left one also, there would be certain traits which would be in dominant form for either A, B or C, right? So everyone would be in dominant form. Only one is in recessive form. What we can do is we can minus this, there would be how many 64 genes which would be formed here. And out of 64, only one is the one which would be recessive one, right? Recessive phenotype. So what happens is 63 would be the one which would be present in dominant form. In either for A, either for B or for C. At least for one of these genes, it would be in dominant form. So the ratio here would be 63/64. So this would be the correct answer. You have to just minus the recessive one. Did you understood? Did everyone understood this particular question, or you want me to repeat it? Or you got confused or something? Firstly, you need to calculate, you need to have an idea what would be the phenotypic ratio for the trihybrid cross, right? There is a simple formula through which you can calculate the phenotypic ratio for dihybrid and for the trihybrid cross. If you know the ratio, phenotypic ratio for the monohybrid cross, right? You just have to do the cube root, take the cube root of 3:1, right? For you can either split it, because we know that the phenotypic ratio for the dihybrid cross is basically 9:3:1, and we can multiply it 3/3:1, right? Then the phenotypic ratio for the trihybrid cross, it could come. And we have seen it for monohybrid cross, whether it is dihybrid cross or for trihybrid cross. Only one trait would be there, which would be present in homozygous recessive form. Rest everyone would be either in either both of the traits would be present in dominant form, or either one of the trait would be present in dominant form. So dominancy would be shown in all of the traits. Only one would be the one which would be present in recessive form, right? Both of the traits would be in recessive form. So here it is being asking that what is the probability of the progeny which shows the dominant phenotype for at least one of the phenotypes controlled by genes A, B, and C, right? One should be dominant. So rest everyone would be showing the dominancy. Only one would be the one which would be present in homozygous recessive form. That only the thing which you need to minus from the rest of. Then the answer that would be coming, it would be 63/64 would be the ratio. Yes? So till here, have you understood? So now here are some other questions that you need to solve. So these questions we'll be solving, and we'll be coming across in the next session, which would be held on Tuesday maybe, right? With unit 13, I will be coming, and there I will be discussing all these questions. So whatever discussion we had in today's session, the rules for the probabilities that we applied, all these things would be applied in these particular questions, right? So firstly, there would be addition, multiplication rule also that you have to apply, and apart from that, the binomial expansion also that you need to know about the Mendelian genetics, right? So here are the two questions that you need to solve, and it would be the homework for all of you. And I will be coming on Tuesday maybe. Then we'll be discussing both of these questions, right? So I hope this session would be helpful for you all. You would have realized what would be the standard of CSIR NET paper related to genetics, like what kind of questions they majorly give in exam, and how through simple tricks you can you can utilize those tricks, and you can solve those particular questions. So I'll be signing off now. And the free sample papers for this particular chapter, this particular unit, you can download through the link given in the description. It would be our Telegram group link. So if you click on that link, you'll straight away go to the Telegram group, and there the sample papers would be present, and you can download it from there, right? Thank you very much.